Welcome to my video on prokaryotic gene expression. So how do we control what proteins are made in bacteria? We're going to use something called an operon. Before we get into operons, I want to review something we did when we talked about controlling blood glucose levels and childbirth, when we talked about feedback systems. Feedback systems aren't just something that happens in our endocrine system. Feedback systems are used all over the natural world, okay? In this example, feedback systems are going to help control whether genes are expressed in these prokaryotes. And we're going to basically be talking about negative feedback. Okay, so let's go through this example. Let's say that we want to make an enzyme, or actually it's an amino acid. We want to make an amino acid called tryptophan. Tryptophan here then is right here, is the final product. Okay, so we want to make our tryptophan. This is our final product. Okay, and like I said, tryptophan is an amino acid. So we have our precursor molecule. Our precursor molecule goes into enzyme 1, and we turn our precursor molecule into this next molecule right here. This molecule goes into enzyme 2, and enzyme 2 turns that molecule into this molecule down here. This molecule down here fits into enzyme 3, and it's finally turned into tryptophan. So just a reminder from the beginning of the year, this is known as a metabolic pathway. You have to go through many steps to take this precursor molecule and turn it into tryptophan. Okay, but what happens when you have enough tryptophan? We want to shut this pathway down. To be most energy efficient, we're going to shut down enzyme 1. So one of the things that can happen is, for example, this tryptophan itself can fit into an allosteric site of this enzyme and inhibit it. This is known as allosteric inhibition. This is also known as feedback inhibition, when the final product inhibits the first enzyme so we don't follow this pathway any longer. Another thing that can happen is, so this is regulating enzyme activity using the end product. We can also regulate enzyme activity by controlling whether or not the enzyme is made at all. Okay, so for example, there are genes TRPE, TRPD, TRPC, TRPB, TRPA that make all of these enzymes. TRPE and D make enzyme 1, or code for enzyme 1. TRPC codes for enzyme 2. TRPB and A codes for enzyme 3. So what if we blocked this gene from being transcribed? Then we wouldn't make enzyme 2. So we would go precursor to this first molecule, but we couldn't go any farther in this pathway. So once again, this is an example of inhibition. This is inhibiting by preventing the enzyme's production that would help with this pathway. This is inhibiting by just blocking the first enzyme. These are two pathways organisms have that they can use in order to inhibit the production of this final product once we have enough. And this is an example of negative feedback, right? Because when you have too much tryptophan, you're shutting down this pathway. When you don't have enough tryptophan, you'll turn this pathway back on. Okay? Now, how is this controlled in bacteria and prokaryotes? We use a system called an operon. Okay? Operon are genes with related functions that are grouped together. Okay? And we're going to look at two examples here. We're going to look at the trip operon, and we're going to look at the lock operon. The trip operon is an example of something called a repressible operon, and the lock operon is something called an inducible operon. There are many different examples, but we're just going to use each of these as kind of a classic example. Okay. In an operon, okay, like I said, there are genes with related functions that are grouped together. Other important words to know are that there's a promoter region, okay, and this is where the RNA polymerase binds. There's an operator. This is where this is the DNA binding site or the repressor protein binding site. And then, not on the operon in a second location, there is a gene for a repressor protein, and this is a regulatory protein produced by a different gene. So these are some of the key words, and I want to go over one example of an operon with you. We're going to do the repressible operon together, okay? And the example we're going to use is a trip operon. Okay, so I want to color code our operon here, okay? This right here is actually the operon. Okay, this whole section is the operon. This regulatory gene is at a different location on the chromosome. So the regulatory gene is at a different location on the chromosome. All of this stuff right here is part of the operon. Okay, so we have the promoter region. That's where the ta-ta box is. We have the operator, right? And we have the tryptophan producing genes. And these are the genes that make the enzymes to produce tryptophan. Okay? So, how does this operon work? I'm going to give you a code up here for how this operon works, and then I'll explain. In a repressible operon, this operon starts on and is turned off. What do I mean by that? I mean that typically what happens when this operon starts on is that the RNA polymerase can bind here to the promoter region. It can move down these genes, and this right here is all the DNA. All of this is the DNA. Okay. RNA polymerase can move down these genes, and it can transcribe these genes, okay, into mRNA. Right? And so remember, prokaryotes don't have introns, so it's exactly the same genes produced in the mRNA that were in the DNA. So we're going to transcribe our mRNA. The letters, by the way, code for the different enzymes. So these letters code for enzymes. Then we are going to translate that mRNA into enzymes. These enzymes are enzymes that make tryptophan. Remember over here, right? Like the precursor molecule goes in, and then we have like all these steps, and out comes the final product, which is the tryptophan. So this is the default. This is on. Okay? So in the default, the operon is on. Okay? Right? Which means RNA polymerase binds, protein synthesis occurs, and we make enzymes that can make tryptophan. So that's what I mean when I say it starts on. Okay? However, lurking in the distance is this repressor protein over here. Okay? Now, like I said before, remember, this repressor protein, okay, this regulatory gene is located on a different part of the bacterial DNA. this regulatory gene codes for this repressor protein. So usually this repressor protein is transcribed into its mRNA and translated into a repressor protein right here. But the default is inactive. So the default is the repressor protein is inactive. So typically in a bacteria, they're going to be making tryptophan, right? Because the repressor protein is inactive, the operon is on, okay, which means transcription of the DNA can occur, translation can occur. We produce these enzymes that help us make this tryptophan amino acid. Okay, so this is the default setting. But the question is, what happens when we have too much tryptophan? Okay, well, when we have too much tryptophan, this tryptophan comes over and is going to bind with this repressor protein. Okay, so this tryptophan is going to come over and it's going to bind to this repressor protein. So default operon is on. However, when tryptophan is present in large amounts, it binds to the repressor protein and activates it. Okay, so this tryptophan is going to come over and bind to this repressor protein. And now it's going to cause the binding of the tryptophan to the repressor protein is going to cause this repressor protein to become activated. Right? And this tryptophan is attached right here. So now we have an activated repressor protein. And sometimes this tryptophan is actually called the co-repressor, because when it binds to the repressor protein and activates it, it's going to help repress this whole operon. Okay, so what's going to happen now? Well, this activated repressor protein, okay, is going to be able to now fit So as I was saying, right, this activated repressor protein, because the tryptophan bound to the repressor protein,